Maybe you can remember finding out as a kid that we're made up of lots of little stuff. Our bodies are composed of cells, little individual parts that add up to the whole of us. Every living thing is made of cells, single cells in some cases. The discovery of cells and all that has led to is celebrated in a new book by Siddhartha Mukherjee called The Song of the Cell, an exploration of medicine and the new human. Siddhartha Mukherjee is a cancer physician and researcher and an associate professor of medicine at Columbia University. He's also a prolific and successful writer. His previous books include The Emperor of All Maladies, which won a Pulitzer Prize in 2011. This is Siddhartha Mukherjee's third visit to the Jefferson Exchange. Welcome back.
Thank you for having me.
I have often said that if anybody studies an issue long enough, they get it down to the cellular level. Is that what happened here? I mean, you've always been interested in cells and had to know about them in your work, haven't you?
As a physician, that's the basic unit of life and the basic unit of disease. So you can't avoid getting down to the cellular level if you are a doctor.
So the first names that you mention in the book, of many, are Schwann and Schleiden, two German guys in the late 1830s. They were not the first to write about cells, but what did they add to the discussion about cells?
Well, Schwann and Schleiden opened the book. There's a beautiful conversation between them, which is just recorded very slightly in a letter years later. Schwann and Schleiden are lab mates working in Germany, and they are studying respectively plants and animals. And they realize that whenever they look down through the microscope at plants and animals, there's a universality, and all plants and animals are composed out of cells. It's not something that's unique or bizarre about one plant or one animal. It's that every living being in the living world is made out of cells. And so Schwann and Schleiden are sort of known as, they're certainly not the first to see cells, they're certainly not the first to name themselves, but they're first to enunciate something that becomes the central tenet of cell theory, which is that every living being is made out of cells. And that, of course, sets off the whole scientific world on a long journey into the world of the cell.
You share some personal stories, too, from practice and from your own life, and you share the story of your friend Sam and his melanoma, and that story makes us aware, once again, of the many different kinds of cells in our bodies. Some of them have to work together. Some of them basically have to ignore each other. How does T-cell therapy work?
T-cell therapy works in different ways, but one example of T-cell therapy that at least Sam was treated by, T cells are immune cells, and their job is to go sniffing around the body. They're like surveillance drones. Their job is to go sniffing around the body looking for foreign objects hiding within cells. So in other words, that's the job of one kind of T cells. There are many kinds of T cells, but they go around looking for, as I said, they're surveillance drones. And if when a T cell finds a virus-infected cell, the virus is hiding inside the cell, it kills the cell. The problem is that the same T cells, when they go out of control, they can turn against your own body, causing autoimmune diseases like lupus or rheumatoid arthritis or type 1 diabetes, among other autoimmune diseases. So there's always a balance in these T cells. And the way that they're balanced, there are many kinds of ways that the body has put together through evolution, the human body has put together to put T cells in check. And those genes, those proteins are called checkpoints. And it turns out that cancers, like melanomas, actually exploit this system. They tell T cells not to attack them, even though to some extent they're, of course, parts of our own body, but they also have foreign things in them, like they are living in places that they shouldn't be living in. A melanoma that's metastasized to the lung, for instance. There's no reason for that melanoma to be sitting in the lung. Cancers exploit this system, and they basically tell the T cell to turn away, to not look at it. I call it, don't shoot me, I'm harmless.
Uh-huh.
And there is a new generation of medicines that can reverse that cancer's hiding away system, reactivating the T cells, basically to be able to recognize the cancers again. And for some cancers, such as melanoma, these are very successful therapies, so-called immunotherapies or checkpoint inhibitors, because they block the T cells activity to turn away, and all of a sudden, like orange jackets that people wear to say that we're part of the system, don't shoot us, we're harmless. They strip away the orange jackets. These medicines do, and so Sam was treated with one of these medicines, a checkpoint inhibitor, and the T cells began to recognize his cancer and turn against it and kill his cancer. And so this is one of the newest kinds of medicines in which we manipulate T cells to turn against cancers.
That is, I would think, the optimum way to treat cancer, isn't it? To get the body to do what it would normally do and zap the thing that's trying to destroy it.
Yeah, it's very optimal. The only problem is that it's very hard to do that, and often has many toxic side effects. As I said, everything comes in a balance. You can tell the body to stop doing exactly that. The unfortunate thing is that then you have a possibility of unleashing an autoimmune disease, and once you do that, of course, it's very hard to stop that, because that disease is can turn it against itself, and that happened in Sam's case. In Sam's case, the T cells turned against not just the cancer, but also against his normal liver, and that became the problem. We couldn't stop the T cells from attacking the normal liver. So finding that exquisite balance is, of course, one of the goals of immune therapy.
So, Dr. Mukherjee, we were just talking about what happened with your friend Sam, who had melanoma. He was given a type of treatment that was meant to get the body's own cells activated to defeat his cancer, but it ended up defeating his body as well. Contrast his treatment with Emily Whitehead's, a pediatric leukemia patient who you mentioned in the book.
Yeah, Emily Whitehead had a different kind of T cell therapy. In Emily Whitehead's case, her T cells were taken out of her body and then weaponized using gene therapy. You can imagine that they were armed with harpoons on their surface to recognize B cells, and her cancer happened to be a B cell cancer. So the T cells were then grown in the laboratory until they reached several million in number, and they were infused back into her body. She was the first pediatric patient to receive this kind of therapy for leukemia. And her T cells, these weaponized T cells, were incredibly active in her body. In fact, so active that at one point of time, they went on a rampage, and the doctors had to figure out, what drugs to give her to stop that rampage. But they did, and she survived and is still alive today. I mean, her chances of dying from this disease were nearly 100% had there not been this really medical miracle, which allowed her T cells to become basically active against her cancer. This kind of therapy, unlike checkpoint inhibitors, is called CAR T therapy, C-A-R-T therapy, and has been very useful in diseases such as multiple myeloma, lymphomas, and leukemias, basically blood cancers. And Emily still survives today, and she is a young woman applying to college, and she wouldn't be alive had there not been this incredible, as I said, a medical miracle that allowed her T cells to attack her cancer.
You tell us in the book that you've watched T cells under microscopes. It sounds exciting.
T cells are amazing. I'm an immunologist as a graduate student by training, and so they're kind of amazing to watch. They come in many kinds. So I should say the ones that I especially have been working on or have studied are the T cells that are killer T cells, and these cells have an incredible ability to, as I told you, sniff out a cell that has a virus inside it, and then they move around. They have little. It's almost like they're walking around on a petri dish, and then they go in and attack virus-infected cells. They give it it's interesting to watch them. They sort of put out a little foot and sort of tickle the cell and see, oh, is there something wrong with it? And then they get activated if there is, and then they start becoming highly active. They become sort of, they change their shape, and then they grip the other cell and throw poisons at the cell so that the cell is killed, and with the cell, the virus is also killed. They're kind of amazing to watch under the microscope. You have to be very patient because this process takes some time, but they're just incredible cells.
The verbs that we use to describe some cells in the body, like the T cells and, putting out a foot and attacking. And we talk about how the sperm cells, they're swimmers and they know where to go and all that sort of thing. We give such agency to cells. They don't have any independent thought, obviously. But, how do we explain the way that cells seem to know what their jobs are?
So, yes, it's important to know that they don't have thought or sentience. But they have agency and function, and that's what makes them special. That's what makes them living. They have the capacity to do things. They integrate information and take that information and convert that into function. They have immune cells have a capacity to detect invaders, and it's one form of agency. Again, it's not sentient agency. It's not like they're thinking that, here's the invader. But they have molecules on their surface and inside them that gives them what I would call functional agency, the capacity to do things, to be actors, to be doers, in a way that nothing else has, because their cells are the smallest autonomous living units. And it's their function that ultimately determines the function of the human body. And it's their dysfunction that determines when the body falls into disease.
We are talking to Siddhartha Mukherjee on the Jefferson Exchange about his latest book, The Song of the Cell, an exploration of medicine and the new human. You dedicate the book to Rudolf Virchow. Why is the book dedicated to him?
Well, Virchow was one of the greatest pathologists who ever lived, an immense person in the field. And I remember we were talking about Schleiden and Schwann in the beginning of the conversation. Schleiden and Schwann were, laid the basis of cell theory by saying, all living organisms are made out of cells. But Virchow made three more audacious statements. So first he picked up from some people who worked before him, which was all cells come from cells. Now, again, it might seem quite obvious to us today, but it was not obvious in the late 19th century. But every cell in your body comes from one cell, the first cell that you were born with, which is the fertilized egg. And that's an absolute truth. So every cell in your body, the neurons in your brain, the cells in your liver, the neutrophils, the T cells, the B cells, every one of the several billion cells you have in your body comes from that one cell. And so Virchow was the first to really realize that the only way cells arose was through the division of prior cells. So suddenly, we began to understand the body as if it was, the development of a cell into a full body. The second thing that Virchow concluded was also very important in its time. We assume it as something that we know already, but at its time was a very powerful and audacious statement, which is that that normal physiology, what we consider, the way the heart functions, the way our kidneys function, is really a consequence of cellular function. So normal physiology is really cellular physiology. And if you want to understand normal physiology, you want to understand how the heart functions or the brain functions, you have to look at how its cells function. And only that can let you understand how the whole body functions. The converse was his last statement, which is that the converse is that if normal physiology is cellular physiology, then pathology, illness, is a dysfunction of cells. Somewhere along the road, there has to be a dysfunction of cells in whatever form or whatever cell it might be. And so these three statements, building on Schwann and Schleiden's earlier work, laid the foundation of modern cell biology. And I find him a fascinating character because he was one of these people who was, of course, an incredible cell biologist, but also a very humane doctor. He was, right through the end of his life, was a huge champion of equality in medicine, equity in medicine. He resisted all the pathological forces of his time. This was a time in Germany when Germany was becoming progressively anti-Semitic. Virchow was a big resistor to all of this. So there's so many qualities of him as a doctor that I admire, ranging from his piercing insights into cell biology all the way to his humanity as a physician. And I thought that, that would be, he's sort of one of the characters in the book that sort of really lead the frontier of medicine and biology and cell biology in particular.
This is the Jefferson Exchange. I'm Jeffrey Riley. Our guest is Dr. Siddhartha Mukherjee, who is a cancer physician and researcher, associate professor of medicine at Columbia University, author of many books, including the Pulitzer-winning Emperor of All Maladies. His latest book is called The Song of the Cell, an exploration of medicine and the new human. This is a conversation you are welcome to contribute to. Your conversation topics, your questions can be sent by email to jx@jeffnet.org. You bring up another case in the book, the case of a patient you identify as MK in Boston in 2002 when you were there. And you tell us that you went back to Virchow for inspiration and treatment. How?
Well, Virchow's idea was that, as I said, that, disease is cellular disease. So if you want to understand a disease, you have to understand what cells there are responsible for the disease. MK's case was very complicated. He was a very complicated immune deficiency. He was a young man, chronically infected by all sorts of bacteria and fungal infections, viruses, and everyone was having a real hard time figuring out what was wrong, because it seemed that, his entire immune system was in disarray. And there's a name for this, it's called severe combined immunodeficiency, but it comes, or SCID. Mm-hmm. But SCID has many, many different forms, or many different causes, cellular causes. Sometimes it's one cell that's causing the collapse of the entire or multiple parts of the immune system. Sometimes it's multiple cells that are causing problems with the immune system. So going back to Virchow that night while I was trying to figure out that case along with lots of other people, we decided to go cell by cell and ask the question by looking at his blood. There are all these modern techniques by which you could do that, and ask the question: what was the central problem? And I often, I liken it to, cells operate in networks. The immune system is a network of cells communicating with each other. So cells operate in networks, and so you have a situation in which you don't know. It's like a fisherman's net. And all of a sudden you see it sagging, and you don't know why it's sagging. Which piece? Where's the broken rope? And the only way you can understand the sagging net is by going, rope by rope, as it were, cell by cell, in the case of MK, and figuring out where the real tear is. And in his case, it turned out that there was one particular cell that was the problem, and everything else, the whole net, or the immune system's net, was collapsed because that one cell was the one that was not functioning. And until you do that exercise, until you figure out where the source of the problem is, the cellular source of the problem, you'll never be able to diagnose diseases and never be able to potentially treat them.
You mentioned an internist in that case who you, not working on that case, but who you thought of during that case, who had urged you to sniff patients. Why?
Well, this was a great internist, a great eccentric internist. And he would say, people, when you look at a patient, you have to look at the patient in full. A physical examination, a detailed physical examination, sometimes is, you have to do things that are that you can find in a physical examination that you wouldn't find otherwise by using all your senses, not just a stethoscope, but your eyes, your ears, hear the patient's voice. You can diagnose someone with, for instance, early Alzheimer's disease by listening to them because they may be missing parts of speech. They may be mumbling in places. They may be replacing nouns, stuttering, for instance. You can diagnose a patient with Parkinson's disease, early Parkinson's disease, by looking at their face, and there's something just undescribable about a kind of stillness that falls across the face of a person, and you can figure out that that's a sign of early Parkinson's disease, often even before they have a tremor. So this incredibly accomplished elderly, somewhat eccentric physician used to say, also, don't forget to use your nose. Smell a patient. You might smell alcohol in their breath, which they won't tell you about. You might smell, there's a particularly sweet smell of ketones that happens in patients who are in a diabetic crisis. There are patients who have other diseases where they excrete particular forms of chemicals, and by smelling that patient, by sniffing, you can tell the disease even before you send off complex case diagnostic tests. It was just an example of how medicine. You're not just using a stethoscope, and you're not just using complicated tests. You're using all your senses to get a whole picture of a patient, and that's why my internist preceptor, my teacher, used to say, "Don't forget to smell the patient."
Wow, that's an amazing part of the book. Not that closely related to the cell talk, but you do take us at this point in the book deep inside the cell and talk about the membrane and the ways in which things get past the membrane, because obviously they need nutrition. You say that the nucleus is the most mysterious of organelles. As these are things that act like organs, but obviously they're organelles because it's in a single cell. What is it that's mysterious about the nucleus? What do we want to know that we don't?
Well, we want to know, the nucleus is obviously not found in bacteria, but found in other organisms like us, like humans. It has a membrane, its own membrane. But what we don't know about it is that the nucleus is where the genome, DNA, our instruction code, our manual, is stored. We know all of that, but we don't know several other things. We think that the nucleus, like the cell itself, has an anatomy, almost like a skeleton, but we don't know how that skeleton is organized. We don't know why certain parts of it live where they do. We don't know how some parts, obviously genes are regulated; they're turned on and off by other genes, by other proteins. We don't know how exactly that happens. So there's an organization to it, much like there's an organization to the whole cell that still remains a little bit mysterious. There are pores in that membrane of the nucleus. We know some things about the pores. We know how they transport molecules like RNA that come and go from the nucleus, molecules like ATP that supply energy to the nucleus. But we don't know exactly, what exactly those pores are doing, how they work, how they operate, and potentially how we could use them as druggable targets by using a new generation of drugs. So the nucleus is sort of the captain's hull of the cell, if you think of this cell as a spaceship. But we still don't know many things about the captain's hull.
So obviously cells, we know, create new cells by division. It's a natural process. I've always wondered, though, what triggers it? I mean, why do cells suddenly decide to divide? Or is it just on a regular schedule or some accumulation of something that makes it do it?
Well, it's a highly regulated process. Cells don't just wake up one morning and decide to divide.
Okay.
Think about, one of the questions I often ask my own students is, when you have a wound, you cut yourself, the wound repairs itself through the division of cells, skin cells, the cells that lie underneath the skin, but then it stops repairing itself. Otherwise, we'd be, why? I often say, well, why aren't we like a tree? Why don't we grow a new branch every time? And it's because the division of cells is a highly regulated process. Cells accumulate an enormous amount of information before they begin to divide. They need signals from their environment to begin to divide. Those signals come in a variety of forms. They may be because the entire compartment of cells has been depleted. So, for instance, if you give someone radiation, for instance, and you kill all their blood cells, then the stem cells, the blood-forming stem cells in their bone marrow will suddenly wake up and realize, again, not because they are magical, but because there are very particular signals that they respond to, in which case they realize that there are not enough blood cells, and they will start making blood cells. Some of these signals come from the environment. Some of them come from very local signals, as in, where the cells are touching each other. Once they touch each other, often cells will stop dividing. And the process is highly, as I said, highly regulated. It's almost like a soldierly deployment, and it goes through phases. First, the cell readies itself for starting division. Then it makes 2 copies of its DNA, so that the 2 cells that come out of the division get 1 copy each of every gene. Then it finally undergoes. There's a very interesting pause after it's been divided, which is a pause to check the integrity. It's sort of like the cell saying to itself, "Is everything okay? Are we all okay? Has there been some damage or some catastrophe that has gone unnoticed?" And then finally, when that's cleared, it actually splits. It divides itself through the middle and splits into 2 cells. So it's not as if the process is random. It integrates a lot of signals. The metabolic or nutrient capacity of an organism, whether cells next to it have started touching each other, whether it's in the right environment, there are hormones that come into play. So it's integrating all of this information and finally making a decision to divide or not to divide.
So the example you gave that you'd use with your students, why don't you grow a new branch or something like a tree? I mean, when cancer emerges, is it because the regulatory process that you're describing breaks down?
That's precisely the point. The regulatory process that prevents cells from knowing when to stop dividing has broken down in cancer, starting with mutations and then cascading into all the signals that normal cells use to stop dividing. Those processes have been broken down. And then I often liken it to a car in which either the accelerator has been permanently jammed or the brakes have been permanently snapped. A normal cell is a car with an accelerator and a brake system that's intact so that it can integrate all the information I was telling you, just like a car does, right? So when a car moves, it's integrating, or when we operate a car, we're integrating information. Is there a green traffic light? Is there a car right in front of me? Is there a passenger in front of me? How fast should I go? But if that same car, if the accelerators are jammed or the brakes are snapped, then no matter if there's a red light, it's going to go through it. And that's somewhat, I'm using a loose analogy, but that's somewhat what happens in cancer because of mutations in genes that regulate cell division, in genes that regulate response to all of these different stimuli are either broken or they're dysfunctional, and therefore the cancer cells keep dividing, and dividing also in places that they're not supposed to divide. So in metastatic cancers, you can have a cell from melanoma, skin cancer, which is dividing rapidly in your lungs, for instance. So that you've got it spot on. That's the process that goes wrong with cell division in cancer.
We have an email question from Peggy in Eugene who writes, I wonder the extent and understanding of the connection between cancer or maybe other diseases and the emotional body, genetics, and, of course, diet. Thank you, says Peggy in Eugene.
So let me start with diet first. This is something that we, as a lab, this is something I'm actively exploring. We have an extraordinary trial combining diet and a particular targeted therapy, and that trial is ongoing for many cancers, including endometrial and pancreatic cancer and breast cancer. What we know about diet is that it's not just simple. It's not like, there's no simple answer like eat goji berries and you won't get cancer. The relationship between diet and cancer is complicated. We know that cancers have a different way of utilizing sugars. Now, that doesn't mean that sugars are feeding cancers. It means that cancers have a different way of metabolizing sugars than normal cells. And that's a potential target for cancer therapy. We've just not been able to do it very well. We also know that some cancers are highly dependent on signals that arise from hormones like insulin. Again, that's a potential way to target these cancers. But these are all in their infancy. Just one last thought, obesity, very strong link with some cancers. But it's not simple. It seems to be a neurohormonal or inflammatory problem rather than just sort of the cancer feeding off the nutrients of the body. Although cancers do feed off nutrients, but they don't do that in just a simple way in which you can say, I'm going to cut out the sugar in my diet and therefore starve the cancer. It's not as simple as that. So that's the diet piece of it. There's no magic diet, but there may be dietary interventions that might ameliorate the way cancers grow and develop. As far as emotional status is concerned, multiple studies have tried to track down whether, there's some trauma or emotional status that contributes to cancer, and they've not really found very much. We know that stress is related, but when I talk about stress is a very loose word. What I'm talking about is a particular form of stress, which is inflammation. We know inflammation has a very strong link to cancer, but inflammation and emotional stress are completely different things. Emotional stress, of course, it helps with healing because you're motivated to go and get the therapy that you appropriately need for cancer or to get a prevention, a screening. But this idea that somehow cancer is a result of emotional stresses, I find very problematic because it's sort of like blaming the victim, and it really causes more emotional trauma in cancer patients when you say, oh gosh, it was your emotional state that caused cancer, because it just makes them feel even worse about themselves. So I don't believe, and I don't think there's very strong evidence anywhere to suggest that emotional stress, the way we understand it, causes cancer. I'm making a very important distinction here, saying that the word stress is used very loosely, and inflammatory stress or other forms of physiological stress,
real physical
have
stress,
certainly been linked to cancer.
Mm-hmm. Thank you for the question, Peggy. And then Gary in, not sure what town from, says, aging of the individual must be reflected on the cellular level, but how? How can you distinguish the cells of an 80-year-old from those of a 20-year-old?
Many ways. Many things slow down in a cell in an aging individual. One of them is its capacity cell's capacity to regenerate and rejuvenate. So your stem cells become decreased both in function and in their capacity and in number. Most importantly, there are these things called telomeres, which lie at the end of chromosomes. You can think of them like the little wiglets that lie at the end of your shoelaces, and they protect the chromosome from breaking down. And the telomeres degrade with aging. So there are many, many ways that you can tell an aged cell from a young cell.
All right, thanks for that question, Gary. I'm going to have to skip past some of the stuff about the way that white blood cells work, but I do want to ask about a specific case again, because we've already talked about Emily Whitehead and the great treatment that she got for her usually fatal kind of cancer. Now, you talked about her initial overly strong reaction to the therapy. Is this akin to how healthy bodies reacted to the 1918 flu?
The 1918 flu was very special because, the flu itself was very pathogenic. Mm-hmm. The reason that people died from the 1918 flu was because of the pathogenicity of the flu. It caused pneumonias And pneumonias are when a virus infects the lung, and then you get that infection then sort of snowballs into the destruction of lung tissue, and then also an immune response. So there's certainly some overlap with the 1918 flu, but the 1918 flu was a true pathogen. It was a pathogen. It was the flu that killed, and the pneumonia caused by the flu that killed people. In Emily's case, it was not. She didn't have a pathogen. There was no flu. There was no virus. It was what might be called friendly, although it was not friendly at all, unfriendly fire, which is that when T cells get activated, they start pouring toxins and they go on a rampage. And it was that was Emily's problem.
All right, yeah, I was drawing the comparison in cytokine storm. Wasn't that part of her situation?
Yes. So as I said, there's an overlap here. The cytokine storm is the T cells going on a rampage, but in classical flu, the problem is the destruction of tissue and the killing of tissue that causes the main problem. There is a cytokine storm that accompanies it, and we now know that some of the pneumonias that are caused by flu can be dampened down by the cytokine storm. But the difference is that in one case there's a pathogen that's actually destroying cells and destroying tissue. In Emily's case, it was the T cells causing a cytokine storm without the destruction of tissue, or at least without the destruction of healthy tissue.
Sid, we got time enough really for one question, about a minute and a half here, and it's about the title of the book, The Song of the Cell. And is this that we know the words but not necessarily the music?
Yeah, there's two reasons. That's the first reason. There's so much more to learn in cell biology. We know the words and not the music. We know how cells exist, what they do, but not how they interact with each other. But the other quick answer is that, the genome, we always talk about the genome as sort of the unit of life. But the genome, genes, DNA, they're lifeless without a cell. So, if you think about our genomes or our genes or DNA as the score of a symphony, it's the cell that sings out that song. And without that song, there would be no music because there would be lifeless. It would be a molecule. So those are the two sort of ideas that are captured in the song of the cell.
Are we somewhat limited by how humans think? I mean, we're sort of like cells that don't have partners or something. I mean, are we not good enough at making educated guesses as groups?
Well, we are. We are absolutely, and there's a whole field of cellular ecology that is emerging. Learning how cells talk to other cells, but it's still in its infancy because there's so many interactions that we have to learn about and capture before we fully understand how the human body maintains all of these complex functions, including its integrity, its immune response, and most importantly, of course, very complex things such as consciousness, sentience, sensation, and thoughts, and this conversation that we're having, Jeffrey.
Well, we have learned a lot, and there's a whole much more in the book that we didn't get to. It's a big book with a lot of stuff, including some illustrations, some by the author himself. It is called The Song of the Cell, an exploration of medicine and the new human by Siddhartha Mukherjee, who is a cancer physician and researcher, Pulitzer Prize winner, associate professor of medicine at Columbia University. Siddhartha Mukherjee, thanks so much again.
Thank you very much.